We are so pleased to have Dr. Joana Almaça joining us from University of Miami. She's going to give a talk titled Pericytes and Vascular Control of Islet Function. Sorry, just letting people in. A little about her. She's an assistant professor of medicine at University of Miami. She's the recipient of the Stanley Glaser Award as well as the Faculty Council Award. And her recent paper came out August 1st, 2023 in Cell Reports. The first author was Luciana Goncalves, and it was pericyte dysfunction and impaired vasomotion are hallmarks of islets during the pathogenesis of type 1 diabetes. So pleased to have you today, and it was great seeing you at the recent NPOD meeting. Your poster was fantastic, and I cannot wait to have you walk us through your work.
All right, Monica, thank you so much for the invitation. It's also my pleasure to be here and to be able to share with you our work. And as I was telling Monica, I think the Sugar Science is a very nice platform for people to talk about science and explain their ideas and recent data in a free way and friendly way. So today I'm going to talk about our work on these cells that you can see here in green on the screen. And you can maybe appreciate that the density of these green cells, which are the pericytes, is much higher in the islet than in the surrounding exocrine tissue. And we think that I want to share with you that our work on how these are the pericytes and how the pericytes control islet blood perfusion and the importance for this control and this vascular control for islet function. So we think this is a novel way, a different way to regulate pancreatic islet function. So what are pancreatic islets? Pancreatic islets are endocrine mini organs that exist in the pancreas, and they are highly vascularized because they need the blood vessels to function properly. It is through the circulation that they sense changes in nutrients, and it's also towards the circulation that they release the different hormones that regulate blood sugar levels. And so islets, they receive blood through feeding arterioles, and these arterioles then branch out into a network of capillaries, and then islet capillaries, they merge with capillaries in the inner tissue. So the islet microvasculature is very well integrated within the inner microvasculature. And now we are learning more about the interaction between these two pancreatic compartments. So you can see here that islet endocrine cells are in very close proximity to the blood vessels. And you see that insulin granules are few nanometers away from the circulation. In the islet, endothelial cells, they are fenestrated. So they have this fenestra. And these also, like pores, also allow and enable efficient exchanges between the blood and the endocrine parenchyma. And we see this close proximity of endocrine cells with the blood vessels in mouse islets as well as in human islets. So there is a lot, they need their blood vessels. Endocrine cells need their vasculature. But most of the studies in the islet biology field focus on the trophic support that the vasculature as well as the extracellular matrix they provide to the endocrine cells. And it is true that vascular cells, ECM, are the source of trophic factors that stimulate the maturation of endocrine progenitor cells into fully mature endocrine cells, which then produce different angiogenic factors and then signal back to the blood vessels to form this complex vascular network. So this is what most of the people focus on. But we think that blood vessels may also have a more active or dynamic function. And by changing blood flow, we can also, and blood perfusion in the islet, we can impact the levels of insulin and glucagon that are found in circulation. And the reality is that there are many studies showing that islet blood flow is a very regulated process by the central nervous system, by circulatory molecules, and also by local mediators. But we don't really know how changes in blood flow impact plasma levels of islet hormones, such as insulin and glucagon. And so we have recently identified pericytes as major regulators of islet blood flow and perfusion. And this is what I want to share with you today, our data on this. So basically today, our presentation will focus on the islet pericytes, on these cells that can control islet blood flow and blood perfusion. And we are going to discuss a little bit their physiological functions and also how they become dysfunctional under pathophysiological states. And so we will explore a little how pericytes regulate islet blood flow and also the impact of this vascular control for glucose metabolism. And then at the end, I would like to show you, as Monica was mentioning, our recent data looking at pericyte function in individuals at different stages of type 1 diabetes. And we are seeing that they are dysfunctional before disease onset. So to first start examining why we think pericytes control islet blood flow, let's first have a look more in detail at the islet microvasculature. So here we have a mouse islet, and you can see here the islet vasculature, which is made of capillaries, made of endothelial cells, here shown in red. They are labeled with an antibody against the endothelial cell marker, CD31. And these islet capillaries are covered by pericytes, here shown in green, labeled with an antibody against NG2, which is a pericyte marker. And you see here how the pericytes they look like. They have their cell bodies, and then they have these cytoplasmic processes that they extend along the vessel length, and they cover the capillaries. And human islet capillaries, they are also covered by pericytes, as you can also see here. And endothelial cells in the human islet are also in close proximity to the pericytes. So the islet microvasculature is made of endothelial cells covered by pericytes.
Can I just ask a quick question there?
Sure, Monica, yeah.
You know, so the kidney also has pericytes, and those are major, contributes to the myofibroblast pool after injury. And how similar are renal pericytes to the pericytes you're observing in islets?
They are similar in certain aspects, they are different in others. So basically, we are now trying to characterize the function of pancreas pericytes, and we are comparing them with pericytes in different tissues, such as the kidney, the brain, the heart, and they share different things, but they are also unique on other aspects. We think that the fact that the islet pericytes is surrounded by endocrine cells, as we will see now as we go in the presentation, makes them a little unique.
Okay, great. That would be really
basically
interesting.
In their physiological properties.
Yeah, to unpack the differences.
Yeah, this is a good question. We will see now as we move forward. So basically, just a brief introduction, what is a pericyte? So pericytes are the mural cells of the microcirculation. Basically, they cover small blood vessels or capillaries. And they have, again, as I showed you, they have this typical cell, like a proeminent nucleus and cell body. And then they have these cytoplasmic processes that they use to wrap, and to give support to the capillary. So they give structural support to the capillaries. They also control endothelial cell proliferation. They are very important to regulate transendothelial permeability. And in different tissues, as I was mentioning, brain, heart, kidney, they also regulate blood flow in these different tissues. But in the islet, as I also told you, we have been focusing more on the trophic function. People have focused more on the trophic function that pericytes have for endocrine beta cells. So Lim or Landsman has shown that pericytes are important for beta cell proliferation, for beta cell maturation, and also for insulin secretion. And they can also contribute to the production of ECM in the islets. But we think, as we see in other tissues, that they have vascular functions. So we think that also in the islet, they may favor and may improve beta cell function by improving the vascular function of the tissue. So we wanted to know whether pericytes have also vascular functions in the islet. And so for them to be able to control, for instance, blood flow, they have to express contractile proteins. And so we first looked for the expression of smooth muscle actin, which is a major component of the contractile apparatus in smooth muscle cells. And you can see here in red that indeed islet pericytes express smooth muscle actin. And you can see it's present in their cytoplasmic processes, and is around 50% of the islet pericyte population, both in mouse and in humans. So they have the contractile machinery. And it was some years ago, Rainer Rodriguez Diaz and Alejandro Caicedo here from the University of Miami, they had already shown that sympathetic nerve fibers that reach the islet, they innervate preferentially these contractile cells. And this was very interesting for us. So we wanted to know also whether these contractile cells that Alejandro had shown are indeed our pericytes. So here we can indeed see that sympathetic nerves, they are present in islets, and they travel along the vessels, and they innervate very closely the pericytes, as you can also see here in this zoom image. So pericytes are the targets of this sympathetic innervation that is present in the human islet. importantly, those that are expressing smooth muscle actin, so the ones that could participate in regulating vasomotion. So this is something that we think is very interesting. And so then we wanted to know whether the pericytes in the islets, they have the capacity to regulate vasomotion and to change blood vessel diameter in the islet. And so to do this, what we have, the platform that we have adopted is the living pancreas slice platform that consists of producing slices from the mouse or from the human pancreas by putting the pancreas in agarose. Then we can slice it in a vibro slicer. And now this tissue is alive. So we can look at the physiology of many different cells that reside in the pancreas. So not only we have the islets, but we have the surrounding exocrine tissue. There are neurons still in the pancreas. We have immune cells. And importantly, we have our blood vessels that are still intact. We have not had to digest anything. And so we can label these blood vessels with fluorescent lectins, as you can see here in red. And if you notice, the lumens are still preserved. So now we can measure and assess what elicits changes in the vessel diameter. So what could participate in the regulation of capillary diameter in the islet. And so we wanted to know whether pericytes participate in these processes. And we know also from the literature and from other organs that changes in cytosolic calcium levels in pericytes are needed for their changes in contractility. So we have used calcium imaging as a readout of pericyte activity. And so basically what we do is we produce slices from animals that express a calcium indicator, a genetically encoded calcium indicator in the pericytes. And we put these slices under a confocal microscope. And then we apply different solutions and we see what they respond to. And for instance, when we apply epinephrine or adrenaline, we see that this increased calcium in islet pericytes. So this is what we do with mouse pancreas. But we also are very much interested in the physiology of these cells in the human situation. And so what we do in human, we don't have transgenic humans. So what we do is we label the pancreas slice from the organ donor. We incubate it with a calcium indicator. So we are labeling all the cells in the slice. And then we identify the pericytes with a fluorescent antibody against the pericyte marker, here shown in magenta. So you can see that our pericytes are with the asterisks. And then we again apply norepinephrine. And we see that norepinephrine is increasing calcium in the pericytes. So now we have a way also to examine pericyte activity in the human tissue. And this is the assays that we have been doing. And so we have seen in several studies that different sympathetic agonists, such as norepinephrine, epinephrine, or phenylephrine, they increase calcium in the pericytes, in islet pericytes. And these increases in calcium are associated with a decrease in islet capillary diameter. On the other hand, we have seen that increasing glucose concentration from a low glucose to a high glucose, this leads to a decrease in pericyte calcium levels. And this is associated with a dilation of islet capillaries. So changes in cytosolic calcium in the pericytes, they accompany changes in islet capillary diameter. And we are seeing that increases in calcium in the pericytes lead to vasoconstriction, and decreases in calcium leads to vasodilation. And so we have performed different experiments in this work, and we have seen that we think now that pericytes, they control islet capillary diameter, and they operate under two different physiological states. So under conditions, upon increases in glucose, we think that the beta cells, they become activated and they release ATP and adenosine, which act on the pericytes to decrease pericyte calcium and lead to a capillary dilation, which will allow a local increase in islet blood flow. But under a different metabolic state, for instance, upon activation of the sympathetic nervous system, we think that sympathetic nerves, they release noradrenaline, which acts on the pericytes to increase their calcium levels, and this leads to vasoconstriction and a decrease in islet blood flow. So this is a process that is regulated by the pericytes, and they change islet blood flow. So then we wanted to know why these changes in blood flow are important and whether they are important. We wanted to know if they affect hormone secretion and glucose metabolism if we manipulate islet blood flow. So this was the next question that we had. And to answer this question, it required a lot of people and very talented people and the work of different laboratories. So Alejandro Caicedo, myself, and then Rainer Rodriguez. And we could count on many talented people such as Alejandro Tamayo, Luciana, and Elizabeth to perform the experiments. So it was a multi-team work to address this important question. And so what did we do? Now we know that the pericytes are in charge, they control islet capillary diameter and blood flow. So now we can manipulate the pericytes to manipulate islet blood flow and see the impact that this regulation has on glucose metabolism. And so we have used two different strategies to manipulate the pericytes. We used an optogenetic strategy, so with light, and a pharmacological approach. In the optogenetic strategy, we expressed a blue light sensitive cation channel rhodopsin, in the pericytes. And so upon shining blue light, this will allow cations to go inside the pericyte, depolarizing the pericytes, which then opens voltage-gated calcium channels, and then calcium goes in, and this leads to vasoconstriction. So this is a way that we have with light to activate the pericytes and constrict blood vessels. And in the pharmacological approach, we took advantage that different cells in the islets, they have different adrenergic receptors. For instance, we have the smooth muscle cells and the pericytes, they express alpha-1 adrenergic receptor, which is coupled to G-alpha-Q. So activating this receptor will lead to an increase in calcium, while beta cells in the islet, they express alpha-2 adrenergic receptor. So activating this receptor will lead to a decrease in cyclic AMP in the beta cells. And alpha cells in the islets, they express beta adrenergic receptors. So now using a drug that only targets alpha-1 adrenergic receptors, we are specifically manipulating the cells that express it, which in our case are the smooth muscle and the pericytes. So we had two ways to manipulate pericyte activity. But we didn't want to manipulate pericytes in the whole body. We wanted to manipulate specifically islet pericytes. So we have had to use a transplantation strategy, which was developed by Alejandro Caicedo, Stefan, and Per-Olof Berggren. And this strategy consists of putting islets into the anterior chamber of the mouse eye. And the islets, they engraft very well on the iris. And so now, and they get revascularized. And we can image by anesthetizing the mouse and putting it under a confocal microscope, we can image the islets as long as the mouse lives. And very important for our manipulations is that the cornea is transparent. So basically, it's very easy to shine light on the islet grafts, and also very easy to apply drugs in the form of eye drops. So this system seemed to be ideal for what we wanted to do. So then we isolated islets from animals that express the channel rhodopsin in the pericytes, and we transplanted them into the eye of diabetic mice. So the animals, before transplantation, they were treated with streptozotocin to kill the endogenous beta cells. So now the islet grafts are the ones in charge of glucose metabolism. And so channel rhodopsin expression is coupled to that of a reporter, TD tomato. So we could see the pericytes that express the channel rhodopsin. And we saw that they remained associated with the islet grafts, which was very good for us because now we could manipulate their function. And as a control, we had animals that did not have the channel rhodopsin in the pericytes. So these ones would also be treated with the light, but nothing should happen to them. So this was our system. We had animals with channel rhodopsin in the pericytes and others without. And so because the islets are in the eye, now we can shine light and see what happens when we activate the pericytes with light. And so this is what we did. We are now imaging an islet graft in the eye, and you can look at the arrow. Now we will turn the blue laser on, and this leads to vasoconstriction. So again, showing that the pericytes control islet capillary diameter. The vessel is open. Now we will activate the channel rhodopsin, and this closes the islet capillaries. So we have a way to open and close islet capillaries in an acute way. So basically, when we turn on the blue laser, we close the vessel, but when we turn it off, we open it again. So we are just opening and closing islet blood vessels.
That's a great model. Very nice.
Yeah, this idea was to manipulate specifically the islet capillaries and see what does it do to hormone secretion.
Yes.
So what we wanted also to look into blood flow. And so we can also do this in the eye because we can inject fluorescent dextrans into the animal, and then we can track the movement of the red blood cells, which appear as shadows in the blood. And so what we again did was to record blood flow, and we saw that we could see cells moving around when the laser was off. But as soon as we turn on the blue laser and we activate the pericytes and close blood vessels, this one closed and there is nothing else moving. And we have quantified these in many capillaries in islets in the eye. And we have seen that activating the pericytes with light and closing capillaries reduces red blood cell flux by 50%. So we have now a tool to activate and to reduce islet blood flow. What is the impact of these manipulations in metabolism? And so to answer this question, we have engineered some cages that are equipped with a blue LED light around the perimeter. And so we have the mice here with islets in the eye freely moving, and they are happy. And we have together animals that have the graft with channel rhodopsin, as well as our control animals. So they are all exposed to the same stimulation protocol. And then we started performing different metabolic tests. And we saw, for instance, that if we start giving blue light pulses to these animals, this leads to glucose intolerance. So in the animals that have the channel rhodopsin in the pericytes. So the animals that we are activating the pericytes and reducing blood flow, this leads to a glucose intolerance, as also shown here in the significantly higher glycemia 2 hours after glucose injection, and also here in the area under the curve. So reducing islet blood flow is creating glucose intolerance in the animals. We also wanted to know what is the impact in the plasma levels of islet hormones. So then we again inject glucose, and then we start giving the light pulses. And here in the control animals, when we inject glucose, we see an increase in the plasma levels of insulin 15 minutes after injection, which is expected. And we see a decrease in glucagon levels. So this is the response of control animals. They increase insulin, they decrease glucagon upon a glucose stimulation. But now the ones that we are activating with light and closing the blood vessels, not only their insulin levels are lower, but now they are secreting glucagon, so which is totally abnormal. This is very different than the response that we have of control animals. And we have calculated the insulin to glucagon ratio, dividing the amount of insulin by glucagon. And we see that activating the pericytes and reducing blood flow is interfering with the expected change and the expected increase in insulin to glucagon that we see in the control animals. So this physiological mechanism, okay, so basically here we've seen with this manipulation with light that the pericyte, these changes in vessel diameter that I was describing you in the beginning, that the pericytes mediate are important for the proper hormonal output from the islets. So this vascular control is important for proper islet function. So I told you that we also did a pharmacological manipulation, and this one involved activating the adrenergic alpha-1 adrenergic receptors using phenylephrine. And so here is just to show that phenylephrine, that pericytes and smooth muscle cells in islets, they express alpha-1 adrenergic receptor. So we have slices again from NG2 GCAMP3 mice, and we expose them to phenylephrine, which is a specific agonist of this alpha-1 adrenergic receptor. And you see here that the islet pericytes, they light up. So they are activated by phenylephrine, as well as maybe some smooth muscle cells. So now this phenylephrine activates the pericytes. We again isolated islets from these animals and put them into the eye of control mice. And now we are applying phenylephrine as eye drops. We want to see whether this phenylephrine crosses the cornea and activates the pericytes. And we see also that indeed these phenylephrine eye drops, they are capable of increasing calcium in the pericytes in islet grafts in the eye. And these changes in calcium in the pericytes are also associated with decreases in islet capillary diameter and reduction in islet blood flow. So it's another manipulation of the pericytes, increasing calcium and reducing islet blood flow that we have. And so then we wanted to see the impact in metabolism, similar to what we have done with the light. And so again, we used animals that were diabetic, and they were transplanted with islets from B6 mice into the eye. So again, the islet grafts are the ones that are in charge of glucose metabolism. And so we performed these experiments with the eye drops 7 days after transplantation, when the islet grafts are still not revascularized. And also we have repeated the same experiments 3 months after transplantation, when the islet grafts are fully revascularized. And so we see that 7 days after transplantation, when we activate the alpha-1 adrenergic receptors, we don't see anything in the levels of in plasma insulin levels. But when we do these manipulations 3 months after transplantation, we see that activating the pericytes and reducing flow leads to a decrease again in plasma insulin levels, which is also quantified here. And this decrease in insulin is accompanied by an increase in glycemia, which is actually what we see when we activate the sympathetic nervous system. We see it decreases insulin levels and it increases glycemia. So we think that maybe the sympathetic nervous system is also controlling islet function by controlling the function of the pericytes and blood flow. So as a summary of this first part of the presentation of the physiological aspects, we think pericytes are important and integral components of the islet vasculature. And they have important functions. They adjust islet capillary diameter and they control islet blood flow under different physiological states. Upon increases in glucose, they allow blood vessels to open up and increase local blood flow in the islet, but they also seem to operate upon activation of the sympathetic nervous system. They are activated, they constrict capillaries, and they lead to a reduction in islet blood flow. And we think that these changes in blood flow, they have important metabolic consequences, at least in animals with islet grafts in the eye. So all these manipulations were done in islets in the eye, and now we are trying to do the same in the pancreas, but it is of course a little bit more challenging. But we think that this vascular control of islet function is very important for a proper islet response and a proper secretion and delivery into circulation of the different hormones, depending on the metabolic state. So this is the conclusion of this first part of the presentation, that control of islet blood flow by pericytes is a physiological mechanism that impacts the hormonal output of the islet. So then we wanted to know whether maybe this capacity to adjust to control islet blood flow is compromised under states of disturbed glucose homeostasis. So to answer this question, we went to analyze and to examine pericyte function in individuals at different stages of type 1 diabetes. And these data have been published, and it was the work of my postdoc, Luciana. And they have published, as Monica said, they have been published recently, in August last year in Cell Reports. And so we have seen that the pericytes are dysfunctional. Not only their morphology is altered, they don't no longer look like the ones in non-diabetic islets, so their morphology is altered, but also their function is compromised. And this is what I want to show to you today. So to be able to look at their function, we are very lucky that we are part of NPODs. And so we have received living pancreas slices produced at NPODs, and they were shipped to our laboratory. And we have received slices from different non-diabetic donors. This is our control group, and from different races and BMIs and both genders. So we have received like 9 different donors, non-diabetic donors. We have also received material from what we called our stage 0 group, which are individuals that have only one autoantibody against GAD65. So they are all GADA positive. And so they already show signs of beta cell autoimmunity, but their glycated hemoglobin or C-peptide levels are normal. So they have no problems with glucose homeostasis yet. So they are our stage 0, our AAB group. And we have also received material from our stage 3 group, which are individuals that already have signs of beta cell autoimmunity, but already their glycated hemoglobin is very high and they have no C-peptide. So they are our stage 3, they are our type 1 diabetic group. And so we have measured pericyte responses and vessel diameter responses in these different slices from these 3 different groups. And so we have seen that in non-diabetic donors, in the majority of these are different, each here is different donors, and each line here is a different pericytes from that donor. we have seen that in slices from non-diabetic donors, there is a big proportion of the islet pericyte population that decreases its calcium levels when we go from low glucose to high glucose. Again, as I had seen, so glucose inhibits the pericytes. And this decrease in calcium is accompanied by an increase in islet capillary diameter. So a decrease in calcium, islet capillary diameter increases. So these are the responses of non-diabetic donors. But now in material from autoantibody positive, we see that there are some pericytes that are still inhibited, but now there is a big proportion of pericytes that are actually activated when we increase glucose, which is totally the opposite what we were seeing with non-diabetic donors. And the average trace actually looks like this. So when we stimulate with glucose, instead of opening up, their calcium increases, and these blood vessels are not capable of dilating. In slices from type 1 diabetics, also the responses are a little abnormal, and they are lower in magnitude. And so capillaries from islets from autoantibody positive, they cannot open when we stimulate with glucose. And also those from type 1 diabetics, they also do not dilate. So as I showed you in the first part of the presentation, in the animals that we do not allow capillaries to dilate, either with light or with phenylephrine, this impacts insulin delivery into circulation. So now this seems to be compromised in these donors. They cannot open up when in the presence of glucose. And this may interfere then with the delivery of insulin into circulation, creating some problems. We looked at the other source of input that I discussed with you today, which is the sympathetic nervous input. And we wanted to know whether the pericytes in these different groups were still innervated by sympathetic nerve fibers. And as we see that they are in the non-diabetic. So you can see the sympathetic nerves innervating the pericytes in the islet. And in islets from autoantibody positive individuals, we still see that the nerves are present in the islet and they are still in close proximity with the pericytes, as well as in individuals that are type 1 diabetic donors. So we see a lot of sympathetic nerve fibers and they're in close contact with the pericytes. So the density, TH density in the islet did not change in these different groups. But we wanted to know whether they still respond to sympathetic input. And so, again, here I'm showing pericyte responses to norepinephrine in non-diabetics. And we see that at a subset of pericytes in the islets, they are activated when we apply norepinephrine. So they increase calcium, and this is associated with a decrease in islet capillary diameter. But now, in slices from autoantibody positive, there are fewer pericytes are activated by norepinephrine, and the responses to norepinephrine are reduced. In type 1 diabetics, some of them are still activated by norepinephrine, but very interestingly, these capillaries no longer constrict upon norepinephrine stimulation. So somehow these pericytes are also, their responses to norepinephrine are also compromised in individuals that are single autoantibody positive and in type 1 diabetics. So they seem to be losing the capacity to adjust islet capillary diameter. But importantly, they are still responsive to other sources of inputs, to other vasoactive molecules, such as endothelin-1. So they are still activated by endothelin-1 and another potent vasoconstrictor, angiotensin-2. So the pericytes are still activated by different vasoconstrictors, but they are losing the capacity to be inhibited by beta cells, and also they are no longer so much sensitive to sympathetic input. And this is just to show other alterations that are present that we found in islets during the development of type 1 diabetes. We can see here that the pericytes and as I showed you in the beginning, are close to the endocrine cells in islets from non-diabetic. So we see this close proximity of endocrine cells to the vasculature. But now in islets with individuals with type 1 diabetes, we see that the endocrine cells are further separated from the blood vessels. And there is accumulation of a lot of things in this perivascular space, of myofibroblasts, the density of extracellular matrix is also thicker. Immune cells also accumulate in this interface. And we think that this may interfere with the communication between endocrine cells and the vascular elements. So this is just to finalize the talk. As I told you, pericytes are part of the islet microenvironment and they cover islet capillaries and they receive input from their microenvironment. We think they receive input from the beta cells, and they also receive input from sympathetic nerves and from endothelial cells. And all these different signals, they act on the pericytes, and they are capable of integrate these different inputs and adjust islet capillary diameter depending on the metabolic state. And these changes in flow, we think they impact the plasma levels of islet hormones. And what we are seeing at early stages in diabetes, before these people have turned diabetic, we are seeing that they are dysfunctional. They are no longer inhibited by the beta cells. They are still innervated, but they are no longer very much responsive to sympathetic inputs, but they are still activated by vasoconstrictors. We also see a lot of accumulation in this perivascular space, and we think that this may interfere with communication between the beta cells and the vasculature. And we think that the dysfunctional pericytes keep the blood vessels unresponsive and capable of changing the flow depending on the state, and this will affect the hormonal output of the islet. So as a take-home message, we think that microvascular dysfunction in the islet may contribute to a loss of glucose homeostasis. And so with this, I would like to thank the people in my lab, Luciana. She has done all the work in the type 1 and also in other experiments. And Katarina, she's a recent postdoc in the lab, and she's working on a very interesting project related to COVID-19 and a potential loss of glucose homeostasis in the people affected with COVID-19. So this would be presented maybe in the future. And also Elizabeth for all the help with the mouse colonies and genotyping and immunohistochemistry. I'd like to thank Alejandro Caicedo for all this help, and also people in his lab, Alejandro Tamayo and Medina, Rainer Rodriguez, Ernesto from our division chief, and important thanks also to people at NPOT for providing the slices and people at the DRI. And thank you very much for your attention.
Thank you so much, Joanna. This is beautiful work, really exciting. And, I want to open it up to questions. There's a bunch of people here. There's some clapping hands in the audience.
Can I stop sharing, Monica? Just I see the faces.
Sure, of course.
Okay.
Yeah, so if there's someone who'd like to unmute here, you can unmute, or you can drop a question. Great job, Joanna, from Alberto Palisi, City of Hope. I'm not sure who's this? Who's instead of Chloe Rackman? Do you want to have a question?
Yeah, that would be great. Can you hear me?
Yes, we can.
Yeah, perfect. That was really beautiful talk. I really enjoyed that. I was kind of interested in the sort of disruption to the pericytes in type 1 diabetes, as in not being close or in proximity to the endothelial cells, as you'd expect. And I wondered if you could sort of comment on, is that just a general physical disruption because of the accumulation of collagen and sort of a general change in architecture, or do pericytes have any migratory capacity?
They have some migratory capacity. We see, like in the ones that we have in the eye, because we have labeled them with tomato. So we see some of them migrating away from the grafts, although the majority stays in the graft, but they do have some migratory capacity. But what we think is happening in type 1 diabetes is that they are changing their phenotype. They are becoming more like a fibroblast, a myofibroblast.
Okay.
And this is also associated with the detachment from the endothelial cells. And maybe this is why then maybe there will be changes in permeability. This loss of pericyte coverage could be associated with an increase in vascular permeability. So maybe we think that this switch in pericyte phenotype, not only we are losing the capacity to control islet blood flow, but it may compromise the barrier properties of the islet. And this is something that we are also very much interested in, assessing the role of pericytes in regulating vascular permeability in the islet.
I was super interested in that in terms of sort of transendothelial cell migration. Do they play a role in sort of immune surveillance? And
they do in other tissues. They have shown in other tissues that they control the immune cell infiltration. They also express adhesion molecules. So they could also participate in this process in the islet, but I don't think this has been examined in the islet. So I think this is something that is an avenue that should be definitely explored.
Cool, that's really exciting. I've got some more questions, but I'll be quiet for a little while.
Okay, thank you.
That's great. Yeah. You know, I just wanted to throw this in here. In 2023, Limor Landsman, which we spoke about her work earlier from Tel Aviv, had this, you know, great study demonstrating the importance of, you know, the local parasitic IL-33 production for glucose regulation.
Oh,
And
yeah, true.
yeah, they support beta cell function and, you know, the pericytes through supporting IL-33. And so when you guys are seeing the pericytes sort of going offline, are you seeing any in your human samples, any changes in the IL-33?
We are not really looking at the immune part of it.
Mm.
Because also we are the amount of material that we get from NPOD is limited. So because it's divided between many different investigators, we have done all these experiments with like 7, 8 slices from each donor.
Yeah.
And it's not a lot to be able to measure cytokine production. And also other labs are looking into those aspects. But I know that Limor has shown that the pericytes are capable, they produce different cytokines. Well, at least they express the gene, the capacity to produce cytokines. I don't know if they has been really fully examined.
Mm-hmm.
But they may have immune or regulatory properties, and I think that should be really explored.
Yeah, no, it was really interesting, that paper. I mean, they showed that the loss of the parasitic IL-33 was even associated with reduced T cell and dendritic cell numbers in the islets or RA production by these actual macrophages that, you know, are locally found there. So the pericytes are really, you know, turning out to be quite important.
I think they're very important, and this is another layer of regulation of islet. The islet hormones have to be, at least insulin, very precisely regulated when it's released. Otherwise, then it does not exert its effects.
Yeah.
So I think that the vasculature can provide this type of control, maybe controlled then by the nervous system. That precise opening and closing will allow these pulses of insulin to occur and then to stop when no longer needed. So this is an additional way, an additional level of regulation of islet function, which we think is extremely important and may be compromised in disease states before actually the onset of the disease. Because if we have a proper vascular function, maybe the beta cells wouldn't have to be attacked.
Yeah.
So we don't really know yet, but we are seeing these changes before onset. So we think they're extremely interesting.
Yeah, it's very interesting. It's really interesting how it's great that you guys are so close to this Casado lab, you know, because they're really digging into the sort of sympathetic innervation of the pancreas. And so it's a wonderful collaborative space down there in Miami for all to work closely.
Yeah.
And I wondered, you know, there was kind of this funny, well, not really funny, but maybe an outlying story from Shireen Forbes in Glasgow in 2020, where they took human umbilical cord perivascular cells they showed that they improved this human pancreatic islet transplant function because they increased vascularization. And so when you talk about that, you know, if you are to like co-transplant those, say, with an islet implant, has anyone looked at what happens to the you know, to the pericytes in that context?
So I think now current strategies to transplant islets and to derive beta cells from stem cells, they are now incorporating that we need additional cells. We need to also derive maybe endothelial cells, and there's these stromal cells, and this will help the survival of the graft and then the function. So I think this is now increasing explored.
Yeah.
I definitely think that if we make the vascular function better, this will help the survival of the cells and their function.
Yeah, it's just another sort of sandbox to explore the pericytes.
To explore and to have into consideration when trying to develop other systems, and also other systems to study the physiology of the islets. I think we should put the different elements, so then we have a picture of how this actually is working.
Yeah, the full physiology.
I don't think we can ignore that these islets are full of pericytes. I don't think this can be ignored, because this is very impressive, I would say. Why would the islets be full of with these cells? I would say they serve important functions, and we are now trying to figure out what. Still, this is all these data are, there are not many people working on pericytes yet.
Well, hopefully this will inspire more. Who else? Is there anyone else in the audience who'd like to ask a question? Feel free to drop something in the chat or unmute yourself. I'm sort of seeing if any hands.
I have a quick question. Joana, do you have a sense of what may be driving these changes in pericytes? And you mentioned that there is perhaps an evolution towards becoming more fibroblastic in nature or function. Do you think there is a way of investigating that? I don't know whether spatial transcriptomic might be handy there. Is there a dedifferentiation or whatever other? What do you think is the process?
Yeah. Hi, Alberto. So yes, so we think that maybe we are trying to we are very much interested in understanding what has triggered this dysfunction in them.
Right.
So we think that the pericytes, they receive input from the beta cells, and maybe there is a problem in this input. So now we are trying to understand because when the insulin granule fuses with the membrane, a lot of other stuff are coming out. We have ATP, adenosine, serotonin, insulin itself, and all these molecules, they may have vascular functions. So we think that there may be something that is altered in this communication, and we want to know whether this is compromised then now in autoantibody and T1D.
Nice.
So this is something that we are exploring, this endocrine vascular communication, because we don't know yet what has triggered their dedifferentiation, but also to understand why they are becoming dysfunctional, we have to use mouse models. So in humans, we are limited. We cannot perform these type of longitudinal experiments. So we have to go to the mouse, and the mouse is not, the vasculature is a little different than the human, but we are trying now to manipulate chronically the pericytes. So if we think that the blood vessels are constricted, and this is why they are becoming dysfunctional, we are using chemogenetic approaches to chronically constrict pancreas pericytes and see whether this leads to their differentiation. And then what is the impact of these manipulations in the vascular niche, whether this leads to immune cell infiltration, to hypoxia, and how it affects then the beta cell function. So we are trying to work on that. Those are the exact two topics that we are now focusing, trying to understand better how endocrine cells talk to the vasculature and whether this is disrupted in the disease conditions. And then what happens if we interfere with this in the pancreas, basically. Does it lead to diabetes or not?
Yeah, I mean, Limor Lansman again in 2021, she's going right alongside you, showed that alongside the endothelial cells, pericytes are a significant source of the islet basement membrane.
Yes.
Which you need for function. So if they go offline, then, pericytes go offline.
ECM is altered, and this can impact then endocrine beta cell maturation, insulin secretion.
You saw some of that in your morphology there. Here's a question from Marjan Slok-Rupinik. Why are all these important functions of pericytes not required in exocrine pancreas?
So the density of pericytes in the exocrine is reduced, although we are seeing that in diabetes, maybe this is changing. We see a lot of fibrosis and a little bit more pericytes in the exocrine, but maybe the differentiated pericytes. And the thing is that the islets are endocrine organs. So their vascular function, this control of blood flow has to be very precise in the islet, because we don't want this insulin to be freely going to circulation. Otherwise, this may lead to hypoglycemia and death. So we think the fact that islets are endocrine organs, they require this vascular control to be really fine-tuned. And the pericytes can provide this type of regulation. Exocrine has a different function. Although I would say they need their blood vessel and they need some type of control, but their function is totally different than the one in the endocrine tissues. If we don't know a lot about this regulation in exocrine, we also do not know a lot. So I think that we are also now trying to see whether the vasculature mediates communication between endocrine and exocrine tissues and how that works.
A lot of room for inquiry. Fad Kadir said, beautiful work, Joana. Would be great to transcriptionally characterize pericyte subtypes across the pancreas. Ooh, that's a cool question.
Yeah, Fad is actually, I didn't mention Fad, but Fad is also an author in this Cell Reports paper. He's a very dear friend and collaborator. And so FAD has actually looked at changes in gene expression in the stellate cell population, where we think that we always get stellate cells from the islets. And we think our pericytes are included in this stellate cell population. And FAD has been able to see changes in different genes related to vascular remodeling, hypoxia, using the HPAP database. He has analyzed those data and is also part of our manuscript. And we are seeing that also in autoantibody positive donors, there is a signature of vascular remodeling in the pancreas compared to diabetics. And this was work done by FAD that I didn't show today, but definitely FAD, we would love to see where the pericytes are within the stellate cell clusters, although it's not been easy to find them because they're not so, basically.
So numerous. Here's a question for Christoph Westenfelder. Thank you, superb work. Pericytopathy has been documented in islets from subjects with type 2 diabetes. What role do intrinsic mesenchymal stromal cells in the vascular niche of islets play?
I didn't, can you repeat, Monica?
Yeah, he said pericytopathy has been documented in islets from subjects with type 2 diabetes. What role, I think, do intrinsic mesenchymal stromal cells, or MSCs, in the vascular niche of islets play? Like basically,
Yeah, so basically
look
the
at these
pericytes,
MSCs.
the pericytes are within these mesenchymal stem cells. I don't know. I know that pericytes are also impacted in type 2, we think. And we think maybe they are also changing their phenotype. We also see a lot of fibrosis, ECM accumulation in type 2, dysfunctional vasomotion. So I don't know if that is attributed only to to stem cells, or to where the pericytes contribute to that. But the problem is that all these cells, they share their niche, their vascular niche. They share different molecular markers. So sometimes it's not easy to say that this is a pericyte, this is a fibroblast, this is a adventitial cell. It has been hard. I think, as Fad was mentioning, it would be really cool if we could really classify these different cell types.
Yeah, you have to dissect the neighborhood. So, and see, what signals are coming from which cell.
Yeah.
Okay, last call for questions in the interest of time. This has been such a great talk.
Thank you.
All right, I encourage anyone and any and all to reach out directly, to Joana Almaça at the University of Miami for future inquiry and collaboration, because it's such an exciting time for this type of work. So thank you again, Joana. It was really great. And we look forward to, next steps and next work coming from your laboratory.
Thank
Have
you
a
so
great
much.
rest of your day. Bye-bye.
Thank you, everyone. Bye-bye.
Thank you.